Hello, and welcome to demola. And today's lesson we are covering stem cells, a really interesting topic in terms of the biology behind it, as well as the real life impacts. So we'll get straight into it. No time to waste.
So we're going to kick things off with a very important definition, and that is what a stem cell is. So a stem cell is an undifferentiated cell that is able to divide and differentiate into specialized cell types. And the reason why I've highlighted divide and differentiate is because it's crucial for you to say that to get your marks, right? So divide and differentiate. In terms of a stem cell, we have types, right? And it's all dependent on their ability to divide and differentiate. So totipotents, they're like up there, yeah? They can divide and differentiate into any cell type. They can form a whole organism. So for example, a zygote, I've got a diagram of one later. Pluripotency, you have a kind of similar thing where they can divide and differentiate into all cell types, except for your extra embryonic cells. And really when we're saying extra embryonic cells, we're kind of talking about like the placenta. So it can't form placental cells, but it can form pretty much anything else. And then multipotent, they're like your lesser ones, they can divide and differentiate into a few types. You're mainly thinking like, like your bone marrow cells can go to like platelets, red blood cell, white blood cell, etc. So I've got this diagram here of a zygote and a blastocyst. I'm going to quickly go underneath to talk about this. So after we have fertilization, right, which we talked about in a previous biology video, you've got the fusion of your haploid nuclei, right? That forms your zygote. So your zygote is going to continue to divide by mitosis and form different stages, different cell types in different stages. So from the zygote, it gets into a molar. A molar is literally just a solid ball of cells, normally like 16 to 32. You form that 3 to 4 days, as we can highlight under here. Cool. Your molar becomes a blastocyst, which is what we've got here. So your defining features of a blastocyst is that it's got a trophoblast and an inner cell mass. And what's really important here to know is that the inner cell mass is going to become the embryo, and it contains your pluripotent stem cells, and the trophoblast is going to become your placenta. Cool. The blastocyst forms 5 to 6 days and will become the embryo like the first week to the 8th week, right? That's when you're in the embryo stage, and then it'll become a fetus, which will be delivered at birth. Okay, here I'm going to be very clear because get this down. Any single question that asks you about gene expression, cell differentiation, you're going to get every single mark every single time. So when we're answering a question like this, you start with the stimulus. And first of all, let's just define what the cell differentiation is. It's literally like a cell becoming specialized. So like you're forming like a white blood cell, for example, from a multipotent stem cell in the bone marrow. Something like that is just becoming specialized. Cool. Stimulus, right? Give an example. So a chemical, temperature, light intensity, anything, there's a stimulus, right? That stimulus causes the activation of specific genes. Cool. Your active genes are transcribed to produce a specific sequence of mRNA. And when I say a gene is active, we'll cover this in a later video about epigenetics, but I generally mean the promoter is accessible, so we'll cover later in the video. But yeah, your active genes are transcribed to produce a specific sequence of mRNA, right? We know what happens to mRNA. We've done protein synthesis. mRNA is translated out ribosomes to produce a specific protein. Cool. Your proteins will determine the cell structure and function. That is the stage we want to get to. So remember, obviously, context of the question, but if you follow these 5 general steps, you will get every single mark every single time, right? And I just wanted to show this. So this is your promoter region. So I'm going to just highlight. This is a promoter region. So that's like a non-coding region of your gene, and this is a coding region. So coding's in green, non-coding's in yellow. And essentially what we need to happen is your RNA polymerase and your transcription factors are going to need to form what we call a transcription initiation complex on the promoter. Once that transcription initiation complex forms, we're more than happy to go and transcribe this gene. So you just need to form that beforehand so that we can actually transcribe, get your mRNA, which will get the protein eventually. I'll be straight up with you. Lac Operon is it's very rare to come up, and I haven't tried this. I believe it did come up in my year, so I just thought, let's cover it in here. So your Lac Operon is normally present in your prokaryotic cells, and we're really going to be talking about E. coli, right? Very common cause of bacterial infections to kind of do with your abdomen and stomach area. It is not pleasant, I'll tell you that. But yeah, what we're really talking about is the production of lactase, right? So your E. coli, they prefer to use glucose for energy, right? Glucose is the best energy store. But when times get tough, right, when glucose is looking a bit low, it's looking a bit peak, they can also use lactose. So that's kind of why we're going to talk about the Lac Operon, right? And Operon, if you ever get asked to define it, is a group of genes with a single promoter region. Cool. So like I said, looking at the next bit, glucose is the best energy source, but it will use lactose if we've got lactose and no glucose. And I've just drawn the Lac Operon here. So let's just talk about definitions. Your P, promoter, your O, operator. So again, we're going to say this is a non-coding region. So non-coding. And then up here, this is your coding region. So like we said, there's multiple genes here. And you'll typically only get these in prokaryotes. So multiple genes, one promoter, right? Lac Z, Lac Y, Lac A. We're going to talk about them a bit later. But when we don't have lactose, what's really happening? There's another protein I've drawn in purple dye, the Lac repressor, right? And your Lac repressor binds to the operator site. When the operator site is bound, it blocks off the promoter. And because it's blocking off the promoter, we cannot transcribe this gene, right? So your Lac Z, Lac Y, Lac A, they aren't going to get produced. That's when there's no lactose. Cool. What about if there is lactose, right? Lactose is going to enter the cell and it's going to form this like allolactose. So allolactose, that's the form it's going to exist in, right? And if I go down here to my diagram, allolactose, this one in orange here, is going to bind to that Lac repressor. And when it binds to the Lac repressor, it's going to do an alley-oop. It's going to head off from the operator site and it's going to leave, right? Cool. Now, if glucose is also absent, we're going to get a signaling cascade. And you don't need to know the ins and outs of it. Just know it uses this molecule cyclic AMP. But basically, we get a signaling cascade, cyclic AMP is involved, and it leads to the production of transcription factors, which are just on CTF. Right? Cool. Transcription factors are made. RNA polymerase is already there. They are going to form your transcription initiation complex on the promoter. Yeah, as we've highlighted there. So when we have that forming on the promoter, we can then say, I'm more than happy to go and transcribe this gene. So you'll produce the Lac Z, the Lac Y, Lac A. Hope that makes sense. If it doesn't, just have a listen back. Now, Lac A isn't really important to be honest, but Lac Z does lactose breakdown, Lac Y does lactose uptake. I just thought would be nice to kind of cover this. Don't stress too much about it because it is rarely ever examined, and I think because it's come up recently, if I'm correct about it being in my year, it's a bit less likely, but definitely just something to be aware of. Like, definitely don't ignore it. Just be aware of what's really going on in the Lac Operon. And now we're heading with something that isn't likely to something that is. So your stem cell ethics comes up quite a lot. There isn't really too much to it, I don't think, but we just need to think about why are we actually using stem cells? Like, what are they useful for? It's really, really promising in research. So they're kind of using it for like regenerative medicine, replacing damaged tissues, treating diseases like Parkinson's and spinal cord injuries, even like motor neuron disease, things like that. Really, really good in terms of doing research and maybe trying to find cures for people with such conditions, right? But there are ethical concerns, right? So we're not here on this channel to debate. In your exam, we're not going to ask you to do a debate, right? But just knowing these things is quite important. So we talk about the benefits. In terms of concerns, whenever anyone uses an embryonic stem cell, it does involve terminating the fetus, right? So you can't take the embryonic stem cells and you'll still get the fetus being viable, right? And that will bring up a lot of religious and moral concerns surrounding the killing of fetuses when it's a person. So that's one thing to consider. Also, embryos can't consent. In the medical field, we're all about consent. It's very, very important. You have to do it before you do any sort of procedure. Embryo cannot consent. The mother can, sure, but the embryo can't. And we're thinking, is that something that we need to be concerned about? Also, these cells have a really big ability to divide and differentiate, right? Uncontrolled cell division is cancer. So risk of tumors is important. Immune rejection is also important. Imagine taking the stem cells from the embryo's terminated, and then the person who receives it can't even, but the immune system rejects it, right? Not great. And also your adult stem cells have a very limited ability to divide and differentiate. So just again, it's not really ideal, and that's why we kind of prefer the embryonic ones. However, you can do a bit of research. Sorry about that popping up. You can do a bit more like tests to see if it's more viable. So I guess that would be a benefit of the adult stem cells. There's actually this really interesting thing called induced pluripotent stem cells, so IPS. But induced pluripotent stem cells, essentially, they're like adult stem cells that you kind of give these pluripotent abilities. I feel like it's less common to come up, and at least it's not something that I learnt during A level. But I definitely think just be aware of it. Okay, we've got three quick questions to try. So first one, you're kind of thinking tissue versus organ. I know we didn't cover it in the lesson, but hopefully it's something that you should have seen at GCSE. Then we're talking about all these like societal changes, concerns. It's a bit extra to what we just said, but just use your imagination, I guess. And then finally about specialization, very common question. Have a go, and then pause the video, and we'll get into the answers. Yeah, so for the first question, you're kind of thinking about like this progression from cells to tissues to organs and then to organ system and then to an organism, right? So if we're talking, so organ system and then organism. So yeah, if you're thinking like this, if you have a tissue, that's just made up of a bunch of cells, right? Your organs are made up of different tissues, and that is what's going to be really a difference, right? And like it said here, you could also just describe it. So like if you said a tissue is a group of cells and you said an organ is a group of tissues, they'd give you the mark. Cool. Now, in terms of your ethical concerns, right? Just first of all, let's go definitions, right? So embryonic stem cells, why are we even using them? Because they're totipotent, right? They're totipotent, so we can form a bunch of different cell types, yeah? Any cell type. Cool. Then we need to think about embryonic stem cells, we're taking it from embryos, right? In terms of what society needs to do, we can't just have people just yanking off embryos, right? That sounds a bit insensitive, sorry about that. But yeah, we need to regulate how we're getting these cells, right? So regulation is really important. Then we need to talk about the moral and ethical issues. A lot of people have problems with using embryonic stem cells, right? So in terms of licensing, really important. Licensing regulation is needed. I get these 2 points kind of similar. I hope you can appreciate that these two are different in terms of 2 and 4. So that one is more thinking about source. This one is more thinking about like what rules, what laws can we put in place that are going to prevent the exploitation of embryonic stem cells. Hope that makes sense. But you're kind of thinking whenever you have to like get management of stem cell use, you're kind of thinking generally about points like this. Cool. And then in terms of your cell differentiation, we've gone through this, right? Like I said, remember the steps. So we said that genes are activated. So genes are activated, and then active genes are transcribed to produce mRNA. mRNA is translated to produce proteins, which determine the cell structure and function. So you can see it's included a lot of those 5 steps. Pretty much all of them, you'd get the marks here. So just stick to that, you will get the marks every time. Okay. So I'm gonna try and wrap this up quickly because I feel like that might have been a long one. But if your A-star tips, quite general, right? So make sure you can say the definition of a stem cell, an undifferentiated cell that can divide and differentiate into specialized cell types. Cool. Potency. Now we're going totipotent, pluripotent, multipotent. Cool. In terms of describing how a cell becomes specialized, really important, quite frequent question to be asked. Normally about 3-4 marks. If you cover those 5 steps that I went through here, you'll get the marks every single time. So make sure that you're sticking to that. And then the more ethical debate surrounding stem cells. I mean, we saw from the clip, from the bit in the lesson, and then from the question that we did as well, there's just a lot of things to consider. So definitely, as we're going through questions, look at the mark scheme, see what's being said, make notes of such points. That will help you out so much when you're talking about stem cell ethics. In terms of what not to do, Lac Operon, like I said, not really common, but if it does come up, you do want to know it. So I definitely say it's definitely worth a skim read before your exams. And then the stages of embryology, like when we're saying that you're going from a zygote to a morula to a blastocyst to an embryo to a fetus, just make sure you can kind of vaguely talk about that thing. And just know about like your definitions as well. So morula being a solid ball of cells, and then your blastocyst having a trophoblast and then a cell mass. Yeah. Okay, that's it for the lesson. I hope it's been very useful. We're kind of wrapping up topic 3 and we're about to go into plant biology. So if it's been useful, please like the video, subscribe to the channel. We're doing really, really well. So thank you all for that. Any questions at all, comment below. I do my best to answer them. I think my response time is fairly decent, so that should be nice. We've got our TikTok and our Instagram, both at A*demola. Follow those, and I'll catch you in the next one. Thank you for listening, and bye for now.